Hello, good afternoon. My name is Maria Gracia Choa. I am going to talk about the DNA replication process. I am going to try to explain as best I can. There are three steps or phases, and they are initiation, elongation, and termination. Also, I am going to explain the process step by step. First phase, initiation. Enzymes that act in the process, the helicase, SSB protein, DNA primase. Helicase. It is the process that separates the strands by breaking the hydrogen bonds between the base pairs. It is also known as the origin of replication. SSB protein is responsible for preventing the strands from rejoining. Topoisomerase copy the same DNA. After the whole process, the DNA starts the primer that is required to bind to the origin. DNA primase. Once the strands are separated, it requires a primer to join the origin. The primers are short sequences of RNA, about 10 nucleotides. Primase synthesis the primers. Second phase, elongation. DNA polymerase III. The builder replicates the DNA molecules to build a new DNA strand. If you read adenine N to the plate, it will only be added thymine T. Can only synthesize new strands in the 5 to 3 direction. DNA polymerases can only produce DNA in the 5 to 3 direction during replication. One chain runs in the 5 to 3 direction, while the other runs 3 to 5. Leading strand that goes from 3 to 5 towards the replication fork is produced continuously because DNA polymerase moves in the same direction. The leading strand, which goes from 5 to 3, the hyper that produced in fragments. The small fragments of Ogasaki in honor of the scientist who discovered them. The main strand is a single primer. The long strand needs a new primer for each of the fragments. Third phase, termination. DNA polymerase I. Once the strand is made, we need polymerase I. Remove the primer and fill in the gaps. DNA ligase. The union of the Ogasaki fragments requires an enzyme to the catalase in and define the gaps. This enzyme is DNA ligase. Terminase once the primers are removed and the ligase has filled in the remaining gaps. This process gives two identical sets of the genes, which will then be passed on to daughter cells. Thank you for attention.